post-human, I think, will be post-biological. By that I mean that it'll be a gradual process. There won't be any sudden transition to a different form. I think we'll gradually integrate more and more technology into our bodies. We'll be replacing our organs with more efficient organs. We'll gradually replace this neural tissue because it dies off after a while. It's easily subjected to chemical damage. So eventually I think we'll replace our brain cells with essentially computerized parts. It'll be much more efficient, much more powerful.
To become cyborgs able to compete with the rapid evolution of computers, we first have to understand how our own brains function. The research in this area has boomed in recent years. One of the factors behind this is the development in scanning techniques. They are now at the level that enables us to observe individual nerve cells at work in living animals.
So is this dying out here? Or is that
it's in the process of growing in.
The technique is based on fluorescent jellyfish. Dr. Lichtman isolated the gene responsible for the fluorescence. He implanted it in mice and continued his experiments until he had a mouse of which only the brain cells were fluorescent.
They're genetically engineered so that these are now heritable strains of mice. We have a yellow mouse strain, we have a blue mouse strain, we have a mouse strain where only a few cells are labeled, and we're now generating mice that have multiple colors in them. We call them brainbow mice.
With this technique, we can see individual nerve cells make connections with each other and emit signals.
It's really quite jaw-dropping. It's beautiful when you look inside and see these nerve cells that have always been there, but never easy to see before in living animals.
This technique is used now by many laboratories, helping in their efforts to understand how the brain functions. Other scientists use a completely different approach. They take the brain cells apart and let them grow in a small dish on top of a computer chip. The problem with this technique is that it is extremely difficult to place each cell exactly on the right connection point. A team of researchers at IMEC has recently found the trick. They printed a pattern on the chip with a product that the brain cells love to eat. While consuming the product, the cells get stuck to the right spot and their tentacles are guided by the pattern. This way, the whole network of brain cells can be entirely controlled. Some scientists have already been experimenting with such brain dishes. Their chips were less precise, but their results sometimes remarkable.
Let me disconnect the light show.
Dr. DeMars, for instance, tries to communicate with his brain dishes and teach them several tasks.
So each of these dishes contains about 20,000 or so neurons, which are firing away as we speak. So each one's an individual network, and they'll fire spontaneously. We take living rat neurons, and they will rapidly form a neural network. And we have this grid of electrodes underneath the surface of these living neurons, and we can listen to the conversation among the neurons. And we can also stimulate activity within that network. We can send in different patterns of stimulation and look at how the network changes as a result of those stimulations. And that's how we do what we do.
What he does is to teach his brain dishes how to control an airplane.
The network can essentially fly the aircraft in a pretty optimal way. So it won't overcorrect too much, and it'll be able to stabilize it in a wide variety of conditions. I think I've programmed like a 50-knot crosswind into this one. It was what it looks like. And you can see it, the aircraft, when it hits one of these crosswinds, how it begins to oscillate. The oldest dish that I've had is at 2 years. And the only reason why that dish died was because we were moving the lab from Caltech to Georgia Tech. Otherwise, that dish would probably still be alive today. Each dish has a characteristic, so you can learn to recognize one dish from another dish. They're demanding, though. You have to feed them once a week.
Dr. Theodore Berger is head of the Center for Neuroengineering at the University of Southern California. With a team of top scientists from different disciplines such as neurology and computer programming, They have collaborated to develop a chip that can replace parts of the brain, or, on a longer time frame, add more brain power.
One of the first things that we do in this process is to actually study this part of the brain experimentally. We need to know very precisely how it behaves if we're going to reproduce that function in a computer chip and put it back into the brain. So what we do is to take the brains out of the animals that we use for these experiments and then slice up this brain tissue and keep the slices alive in a dish. And we study the electrical properties of these neurons. Neurons happen to use electrical impulses to communicate with each other the same way that transistors do in a circuit. And so essentially what we do is to test the cells to find out when we give them computer-generated signals of all possible signals. We give them random signals and we ask, how do they behave? And by looking at the translation, we figure out what the function is, and we can describe that function with a set of mathematical equations.
This portion is simulate one neuron.
My colleagues have taken that mathematical model and built a microchip circuit, very small, millimeter by a millimeter. So we can take a part of the hippocampus that's a circuit, we can cut out parts of it, throw it away, and we replace it with the microchip. And so now the whole circuit works just like the biological circuit, so that incoming information looks like, incoming temporal patterns look like outgoing temporal patterns, just like it does for the biological piece of tissue. But now there's a microchip in there, instead of having biology in there.
The chip is presently being tested on animals. One problem that remains is how to connect the chip to the brain. Today's technology uses tiny electrodes, but in the future, another option might be to put some brain cells directly onto the connection points of the chip. They would then be guided to form connections at exact points within the brain in order to establish the perfect interface.